very quick introduction. So, hello everybody. And you're welcome, Martin. Please grab a seat. So we're very excited to have two pivotal people at the NHSBT coming for our BTRU seminar today. And this is a slightly different take from the kind of academic research studies. This is very much on the implementation of BTRU, which was requested from people who attend the seminar. So we have Amanpreet Dhesi, who's here, and he's Associate Director, Technical and Scientific Development at NHSBT and based in West London. And we have Debbie Rahman, who is a Program Director of Blood Supply. And you rove all over the country, but based in Lincoln as a home destination. And both are very interested in plasma, in post-donation testing, loads of things that cross over our research. And how I came up with this idea to invite, first of all, a man, because I heard him speak at the Cambridge Science Festival a couple of years ago, a wonderful talk, along with Dave Roberts and Mark Croucher. Dave Roberts, you all know, is on the research side of our BTRU, and Mark Croucher is on the marketing side and also involved in some of the projects in our BTRU. So it's a real privilege, and they're going to talk about an inside look at the blood service, which I think is a fascinating topic, and really appreciate your time, and welcome to everybody who's online. So brilliant, thank you.
So just to, although that was a lovely introduction, I'll just reintroduce myself. Yeah. So I'm Debbie Rahman, and I'm one of the programme directors in NHS Blood and Transplant, and I'm responsible for a number of the projects and programmes that fall within the Blood Supply Directorate. So that's from blood donation, collection teams through to manufacturing, testing and hospital service. I've actually been in the blood service for 30 years come January even. And my background is I trained as a nurse in the 80s in Hammersmith Hospital, and then actually came to the blood service as a nurse.
Thanks, Debbie. Right, that's it for Dhesi. So I'm the Associate Director for Technical Scientific Development, which is just a really fancy way of saying we're a team of subject matter expertise scientists, and we support the manufacturing, hospital services and testing teams within blood, as well as some teams, the technical aspects in collections, to ensure they can do what they do. So any of the tenderings, any of the changes, we tend to oversee it. Also, if there's a problem, there's someone in the team that knows everything about something, whether that's a piece of equipment, a particular test, or even how it connects to the system. So we kind of support that. In terms of my background, I think I've worked in or around blood for 21 years. It was not planned. It just worked out that way. I started off by, so I'm a registered biomedical scientist, but there's been many, many years since I've done any lab work. I started off by actually fractionating plasma. I'm going to talk a little bit about that. We'll come back to it. Then I left, and then I worked within some of the labs we're going to show you now. Then I left, and I ended up doing some non-science stuff, where there's projects, change, transformations. And I came back in this role about 2 years ago, and it was almost like a nice circle. It was like I was coming back home as well. And thank you, Moran and Valerie, for the invitation to speak as well. We're just going to start quickly by, we're not going to talk too much about strategy and bore you about NHSBT, but we kind of wanted to set the scene in terms of why we're here. So our strategy, our whole NHSBT strategy, is based around the ambition where we've got a vision where every patient gets the donation they need. So that's the strategy, and I'm not going to go into it. If anyone's interested, it is available online. In terms of an organisation, we're quite complex in sort of thing. I mean, we're going to focus about the blood, but we're quite complex in terms of organisation. So we've got a frontline service to patients, but we've also got a frontline service to donors as well. In terms of our scale, the blood is for England only, but we actually do organs and tissues for the whole of the UK through NHSBT as well. In totality, there's about 6,500 individuals that work for us as well. In terms of turnover, I think we're around 600 million turnover. We do a lot of collaboration as well. And this is just a flavour of some of our labs and some of the works we do, highlighting those that are outside of blood scope as well. But today, we're going to give you an inside look into the blood service. And Debbie's just going to go through some of the key points that we've got, just focusing on blood.
Okay, so some of this information is very similar to the previous slide as well. but just I'll draw out some key blood facts for you. So from a platelet perspective, we treat 69% of our platelets go to people with cancer, 17% post-surgery, 8% treat diseases, and 6% intensive care. And it's around 1% that go to people who've been part of a traumatic incident as well. Blood, we, 2/3 of treat conditions including anemia, cancer and blood disorders, and 1/3 is around surgery and emergencies, including childbirth. You can see that we have around 800,000 active donors, and a small proportion of that is O-negative donors. Big draw on our O-negative supply. We have around 13,500 platelets. And 40% of our donors are under 40 years old, and 45% of our donors are male. Aman went through some of the numbers on the previous slide around our blood donations and our collection centres and our donor centres, and we are currently actually looking to see how many more donor centres that we can put in, and putting them in the right place as well, so that we can attract donors of the future, really. So that's what we're looking at the moment. And again, we have 2 testing sites in Filton and in Manchester, and we have an MSL lab in Colindale, and we have 3 manufacturing sites, again, Filton, Manchester and in Colindale as well. So you can see over 2 million components are manufactured and 29 million tests are completed. So it's a phenomenal amount. There's a hell of a lot of logistical stuff on the other side, making sure that we can get the platelets and the blood and the plasma to everywhere it needs to go. And our transport departments, over 86,000 routine deliveries, and then we have the ad hoc deliveries on top of that, as well as an emergency, and lots of vehicles. It's a big organisation, just on the blood side, and then there's more to it in the other areas.
So in terms of blood, has anyone here ever been to hospital and seen a final unit of blood, or has received a final unit of blood? You kind of see this, it's a finished product, fully labelled, it just comes out of the hospital and it gets administered. But actually, there's a lot that goes behind it, and we're going to cover that. But before we do that, we're just going to put the patient at the heart of what we do, because the patients are the heart of what we do. And this is just a Cambridge study from Salome. She's a sickle cell patient who receives 8 units of blood every 5 weeks, and she actually gets her therapeutic service from NHSBT as well. Now, she's an RO patient, so that's her blood group characteristic, is an RO. And in terms of have we got enough RO blood, which is one of the blood characteristics we've got to supply it, as an organisation, we're only meeting about 50% of the patient demand for RO. So actually, there's a lot of gaps we have to do. So what we're doing is, rather than not supplying anything, we're giving an appropriate alternative in O-neg. So that impacts on our O-neg stocks. And O-neg is a universal group as well. So that's why it's even more necessary. Now, with sickle cell, it's a lifelong disease. It's treated through therapeutic. So, it's treated through exchange transfusion. That's the word. It's treated through exchange transfusion. Now, there has been someone who has successfully overcome the disease from gene therapy, but actually gene therapies are expensive, they're not readily available, and they're not licensed as well. So there's still a lot we have to do to ensure that we can support patients like Salome with the blood she needs.
Okay, so from a donor perspective, as well as our patients, our donors are exceptionally important to us. And I know there's a number of donors in the room here today, but if you come to donate, you will get different experiences depending on whether you go into a donor centre, whether you go into a plasma site, you're giving platelets, or you're giving whole blood in a village hall, and it can be quite different experience depending on where you're donating. And we have absolutely a phenomenal, wonderful donors who keep the service going. And this is just one story of thousands of stories. But this is a really nice one where Molly has a Brom donor card, and she's having donated for 5 times, but she comes from a family who've been donating, and that is really important in word of mouth, encouraging donors. A lot of our donors donate because they've got family members who have donated, or they've got family members who've received blood. Her grandmother, Judy, has made over 100 donations of own egg blood, and I think she's 71. So she's been donating for a long time, and it's absolutely phenomenal. They love getting, they've made a quote there about getting emails showing you where our blood ends up. So I don't know, those of you who are donors, if you've received a text to say your unit of blood has gone to this hospital, and it's really good, warm feeling for our donors. But it's a lovely story where they're exceptionally important. They've given their time and their energy and their blood over a number of years, and they're carrying that through. So as well as our patients, it's really important to recognise our donors in this story. Okay, so this is a very bad infographic, but I'm not artistic. But we just wanted to show you, because this was about the backstory to donating, we wanted to show you what is the donor journey. And currently, if you go onto a session, and hopefully those who donate today, you'll be able to say this is what you are experiencing. You will come to the front of house, whether that's in a donor centre, whether that's in a mobile team, and you'll be met by someone who will kind of check you in at that beginning of that service, make sure that you've got your donor safety check, and ask you to drink an isotonic drink just before you donate. Then you'll go into a waiting area. You probably have brought your donor safety check with you. You'll potentially, that waiting area can be quite a long time, so hopefully not, but depending on what's happening ahead of you in the screening area, it can cause a wait there. You then go through, you are screened in 2 ways. You have your finger prick test to make sure that you've got enough iron on the day to donate, and then you'll also go through a health check to make sure that you're fit to donate. There's no reason for not donating. Post that screening section, you'll then wait a little bit, and then hopefully go through to a donation chair where you'll donate. And then after that, bit that everyone talks about is the biscuits and the cup of tea, and everyone's got their favourite, and if a type of biscuit disappears off a session, sometimes that can cause a bit of angst for our donors. But that generally is hopefully what you're seeing on session currently today. I'm looking at heads to see if that does resonate. It's not a perfect journey, and from a donor experience perspective, the waiting time in particular, especially if you're a first-time donor, can be quite anxious. Some of our donors have, waited over an hour. That's not what we want. We want people to come in as quickly as possible on the whole blood side, donate, have a good experience, and leave in a healthy way. So we are looking at what we can do to make that experience much better from both a donor perspective and also a work perspective. So one of the areas we're looking at is a new donor pathway, and just to show you a little bit of data around why we're looking at that. So if you're a regular donor, your average donor journey time is around 39.5 minutes. It's probably around 38 minutes at the moment. But as a new or returning donor, it significantly increases, mostly because you probably got more questions that need to go through. There may be more reassurance. There's going to be reasons why you're a returning donor and you stopped donating. So that then has a significant impact on the screening time and the wait for all donors. So we're looking at what we can do to look at the new donor pathway so that we can improve that first time donor experience. We can support the understanding of the donation journey. We can give information on different blood groups and different donations and focus on that retention. getting a first donor to a second donor is really important, and a second donation to a third donation is really important. If we can get someone on that journey, they're more likely to stay with us for life. So that's a really important experience. The other areas that we're looking at is off-session screening and post-donation testing. Again, if you look at the screening times for regular donors and new and returning donors, you can see that a regular donor takes around 6.5 minutes, or a new and returning donor takes more than double that time. So again, you see the impact on the rest of the donation session. So what we want to get to is a point where you can come into a session and you can go straight to donation. But you don't have a wasted journey, so you don't get to the session and you either can't give for a medical reason, or you can't give because you failed the copper sulfate finger prick test. We want to get away from the finger prick test. That's probably one of the things that most people don't like. Having a needle in your arm is okay, but a finger prick test just is a no-no. And we also want to give that capacity and more appointments available as well. So that means that we can see more donors. So our future donor journey is very much, if we can get to come straight into session, if we have off-session screening, where most of the deferrals are looked at off session so that we know that our donors are fit to donate, that we just get to a point where someone comes in, has anything changed and you spoke to someone? No, let's go straight to donation. And then we take a sample of blood afterwards to test your hemoglobin, possibly your ferritin levels for the next donation that you come into, so that you don't have to have our finger prick test there, but we still keep refreshments on sessions because we don't want to remove that. And we want to make the donor experience the best it can possibly be. So take some of the unnecessary steps out of a session and really focus on the donor side. So how are we getting to that journey? Well, there's an awful lot of work that our donor experience teams have done in looking at those first steps to off-session screening, looking at how we can make information on our web pages more accessible, and also to answer some of those questions that people get deferred for a lot. So whether they're the travel questions, whether they're the tattooing questions, those sorts of things that stop you from donating. So the marketing team have delivered marketing automation, really driving new bookings, new email system to ONEG donors as well. They've piloted a new website, looking at the content management side of that, and also the careers website. They're doing the first steps in the donor readiness piece. So this is the piece of, that off-session screening piece, new ways to ensure that donors arrive ready with improved eligibility on session. They've got launched the platelet self-service as well. We've had for a long time been the ability to book appointments from a whole blood perspective, but not have the same sort of thing for our platelet donors. They've done a hackathon looking at the whole booking journey, so to make it better for our donors coming through. And then they've also responded to stock challenges and revamping the queuing experience. So an awful lot of work's gone on the front end to make it better for donors coming through the system and make it easier to book an appointment. So one of the pieces of work that I'm involved in is the post-donation testing. I don't know if Lois is in the room or...
No, but Hong Chow, who is on that project, is here? Yeah.
Yeah. Okay, so you might recognise these slides.
Yeah.
Some of them.
Are they yours?
Nice voice. Yeah.
So I've chosen, so Lois did some fantastic slides for us at NHSBT, just trying to really simplify what post-donation testing is about. So I definitely need to recognise Lois for the next two slides that we see. And from a post-donation testing perspective, we really want the donors to be able to come in, as I've mentioned, and have the test after they during their donation, so that they don't have to have that finger prick test. It's a better experience, but also from a health perspective, we get much more data and information on what their actual hemoglobin level is like. And then depending on that, we can say this is when you need to come back to donate. So our current policy is a donor will come through, attend a donor centre or a collection team, and they'll have an on-site hemoglobin test, which is copper sulfate. And we're one of 3 blood services in the whole world that use copper sulfate. So we'll put it into context as well. Depending on whether that they pass that, then the person will then go on to donate, and then they'll come back in 12 weeks if they're male and 16 weeks if they're female, and then the whole process will happen again. If they don't pass that test, then they'll probably have a HemoQ test just to check, just to validate whether that test has failed or not, and then they'll be deferred for 12 weeks. So that's our kind of current process. Where we want to get to is how can we reduce the deferrals and wasted journeys while maximising those donations on session. So our aim is to attend the donor centre, donate, have a blood sample taken, and then that will be tested back in the lab, and it will say, yes, you're eligible to return, and that there will be a kind of an interval about when you're eligible to return. There's a bit more complexity around that, in the sense of, depending on your result, you may then have to come back and have a test on session and donate. But we're just working through this at the moment. And we're hoping that at the end of next year, the end of next year, we will be in the phase where we can actually test this out on collection teams, which is quite exciting, because we've been talking about post-donation testing for quite a long time. So, and it is definitely built on all of the research that we've done in the past with the interval studies and the stride studies. So this is really exciting work, and it will make it much better for collection teams and for our donors. So it's really good. So we want to get to the middle. We want to remove some of the faff from around the edges and really make it a much more streamlined process on session, but a better donor experience, and really focus on the retention of our donors to come back and donate. So that's where we want to get to. And this is a very busy slide, but I just want to show you that as part of the work within the Blood Supply Directorate, we are looking at what we can do to remove touches within the supply chain. So there's a bit there, number 4 is straight to chair. So the work that we're doing to kind of make those processes more streamlined, much better, is number 4. But there's a massive piece of work going on across the whole of blood supply to try and reduce those touches on every part of the system, so that we can put all the value added stuff in rather than an awful lot of the stuff that takes time. So whether it's the front end with our donors and engagement and how we make it much more better. So for example, a digital health check, which is one of my desires before I retire, is that we catch up with the 21st century. That will make a big difference to people. Or whether it's back in the manufacturing or the testing and having more automated processes. So just to show you that the bit I'm talking about on the donation side is part of a much bigger activity that's going through the organisation. On our plasma side, I mean, again, this is another really exciting part of the business. Over the last few years since COVID, has really driven our plasma side. So we're looking at to be more inclusive from a plasma and platelets perspective. The majority of our plasma donors are male. We want to look at technology that will actually allow more women to be able to donate as well. We're building that plasma donor base up, and also to increase that frequency of donation. And there was someone in the room who was talking about the increasing frequency of donation, which is another research programme going on, which is really good. And obviously, more people now are eligible to donate blood based on their health, travel and sexual behaviour following fair recommendations as well. So all of that activity. And then just another thing on enhancing the donor journey. So as we go through a session, there's another piece of research that's going on at the moment or just about to start that Mark Croucher in Donor Experience has been leading on, which is actually the impact of neurodiversity conditions on ensuring that we get the right answers to the questions as we arrive in the session. Sometimes our donor health check may be written in a certain way, or particularly if you potentially have dyslexia, it may not be easy to read. So how can we actually ensure that the questions that we're answering are understood by all? So there's also the Hidden Disability Sunflower Scheme. So some of you may be aware that I'm wearing a sunflower today. Hopefully some of you know what that is, but you know, it's really about promoting understanding that people do have hidden disabilities and making the experience for people coming through a donor session the best it can possibly be. So we have just launched that, or launching that now. And then the other area is the Oliver McGowan training. We are part of the NHS, and the tragic story of Oliver McGowan, who died at the age of 19 due to kind of misinformation at the time and a lack of support. He was a gentleman with autism and learning disability. It's really about understanding those challenges that people face within the NHS. So all of that work is going on to support that donor journey as well.
Thanks, Debbie. I was sitting down relaxing and listening to the talk as well. Okay, so I'm going to quickly talk about what happens once the blood's off the session. Now, for those who donated, you see the blood goes into a pack, right? Actually, that pack is really important. That is it's a medical device, and it's actually an inline system. So I don't know if you guys have actually handled it. I've got a pack here, but what you've got is you've got a series of bags that are connected together, and then you get filters as well. And actually, there's different types of these bags and filters and the combinations you get. And unbeknown to you as a donor, depending on your group, you know, your past blood tests and stuff like that, you're going to get put into a different pack. The experience is the same, but the packs are different. We've got two main pack types, but actually there's varieties of the pack types depending on how many inline filters you've got and where you start with. But the first thing I wanted to say is we have got a pack. If anyone wants to have a look at it, you know, please do have a go. That's our first system that blood goes into. Blood stays within that system until it goes to the hospitals as well. So actually, once the donor's blood's in this system, the end component, it goes into a patient as well. So it all travels through that system and the various things we do. Yeah, okay. That's what we do. Yeah, thank you. So we get these blood packs, various collection systems we have through it, and it goes through our manufacturing testing facilities. We've got 3 manufacturing sites, one in North London, one in Manchester, and one in Filton. Depending on where it's collected, it gets distributed there, it gets processed, then it gets redistributed to 15 stockholding sites, one of which is in Cambridge as well. And we've got the testing sites. I've got some pictures. So the first thing we do with all of our blood is leukodeplete it, which are these filters here. Basically, all that means is we're filtering out the white cells, the leukocytes from the system. We don't do that for all our components, but I'll go into those things. But all of our blood, except for the ones that we want to retain the leukocytes in, is leukodepleted. That's always the first step for most of the pack systems. Then we centrifuge it. Centrifugation is literally just an accelerator of gravity. We could just leave the blood pack as it is, and it will separate out into the component types as gravity cells. But we don't have that much time because some of the stuff is really restricted. So we use a centrifuge just to accelerate that process so we can separate it out a bit more. Then we use a blood separation system. It's basically, once it's separated, we use a system like this. We've got a couple of different varieties. And through a process of pushing, detection, and automatic sealing, the unit from here is separated out into its different components. So a separate component goes into each pack type within the pack system, and then some of it's sealed away. What you broadly get is you'll have a red cell, and a plasma unit. But actually, depending on the pack type, you might have a red cell, a plasma unit, and then a buffy coat later as well. Now, most of them are, pretty much the final product. Once you've got a red cell split up, that's your red cell. All we're doing it is labeling it and sending it to hospitals. Sometimes we pool donations as well. So some of the donations, we get a buffy coat, which is a layer that's high in leukocytes, but also high in platelets as well. But it's not enough to give an adult therapeutic dose to a patient. So what we do is we pool 4 of them together with a additive solution and put it into another bag. And then from 4 donations, we get a single unit of platelets called platelets. Is what we make platelets another way, which I'm going to show you. But one of the ways we make platelets is through pooling. That is from 4 donations. So really, we're probably the ultimate recyclers here, right? See what I mean? Everything we get with blood, we do something with it. At Debbie said we do about 29 million units of testing. So we have about, I think we get 2 million donations through, but actually we screen a lot sort of thing. So we test it for, anything that is at a higher risk of being transfused through blood transfusion. We screen for it as well. And we're quite good with that screening as well. These are just some of the machines. If anyone works in hospitals, these machines are going to be quite typical where you get in hospitals, but we've got them set up for screening. So that's our Alinity machine. We're just bringing some more of those in. This machine here is actually something that screens platelets. So platelets are stored at 22 degrees, which is an ideal temperature for bacteria to grow. And it's got all the right mediums and stuff like that. Now, we bacterial screen all of our platelets, but we don't just do it straight away. We keep them in quarantine, as it were, to allow if there's any bugs to detect it. Then we take a sample for aerobic and anaerobic growth, and we put it through this analyzer. Once it's negative, we then release it to hospitals, but we keep screening it until the end of the shelf life of the platelet. So if there is a detectable growth, once we've issued it, we can let the hospitals know and bring it back straight away. This is really good, and actually the number of bacterial transmissions has completely dropped out once we've implemented this as well. This is just some of the other analyzers, and for the one on the right there, it's we actually pull the samples. So rather than just doing an individual sample test, we actually pull 64 donations into one pool, and then we actually do 3 tests in one go as well. And that's just because the test allows it. So efficiency is important to us as well as safety as well. And this is just an analyzer that we've got for higher throughput as well. So once we've done all those separations, we've done all those tests, we get a range of components. Routine components, I'm hoping everyone's heard of. If not, we can go through. Red cells, they're about 35-day shelf life. We have got a project at the moment. So this blood pack seems simple, right? It's actually got a plastic polymer in there that's actually been banned by the European Union from 2027. So we've got to get a new blood pack made, validated, and components and stuff like that. It's such a hard project. We've actually started an international collaboration on it. As part of that validation, we're actually to see if we can validate red cells for 42 days as well. Platelets, we've gone through. There's 2 ways of doing it. Pools, and then there's component donation as well. Fresh frozen plasma and cryoprecipitate are frozen products, and while they're frozen, they have a 36-month shelf life as well, which is reduced once they're thawed. Broadly, if you look at NHSBT's component portfolio, which is available online, we have about 137 different component variations. Now, if you split that within the 8 main blood groups, and then you split it again with what's irradiated, what's not, and then about, I think we get 1.2 million orders for red cells, about half of them have additional phenotyping. So they don't just want APOS, they want APOS, which is negative for certain groups. So that's a real complex system. Do you see what I mean? That's why we are a healthcare biomedical-led organisation, but actually we're a manufacturing organisation. We need to make sure that the right blood is at the right place and is always available. We do loads of other component stuff as well, where we get into individual novel components, which we don't try to do as a standard, but sometimes for really difficult patients, we do it. We've got a lot of various components. And the reason for that is you can compare it to other blood services. It's not because we just want to be busy. It's actually the more components you got, the harder it is to manage. But we do it working with the clinicians, the research scientists, to make sure the components we have are the right things for the patients. And actually, here's some other components that we've got actually development in the pipeline. I'm going to mention a few of them now. when I drove into your award-winning car park, which I noticed, I think the car park says it's an award-winning car park. As I left there, I saw your air ambulance. I think it was leaving, sort of thing, and I heard that, and I thought, oh, yes, I can bring that in. So we just finished up recruiting 900 randomized control trial patients with an air ambulance, with the air ambulances. So this is SWIFT. This is the study of whole blood and frontline trauma. And this is looking to see if we can bring in a new component, which is whole blood, which is just literally the blood you get leukopleted. So that's going to have red cells, plasma, and some platelets in there as a single component that the air ambulances can take, and they can give it to people who trauma at the roadside pre-hospital. And actually what we're looking to see is if we can improve the survival rates for trauma patients and then actually reduce the need for additional transfusions once they're in hospitals as well. So that has recruited, and that study finished early. It should be reporting in autumn. And the expectation is once that reports, this is then going to be another component we're going to have to bring in, which is really good for patients. It's a huge logistical challenge for NHSBT, because that's another unit where we're taking away other components as well. And it's another thing to add to those as well. So that's going to be something that we're going to do. But we're trying to do more with what we got as well. So another way we collect platelets is through component donation. So someone comes in and they give 2 to 3 units of platelets. At the moment, those platelets are, if you see on the left, the current apheresis platelets, they are resuspended in plasma. What we're going to do is, we're actually trying this, it's just going through a phase 0 validation study before we scale it up, is we actually want to keep that donor donating the same amount. But what we want to do is we want to keep those platelets in some plasma as it's hyperconcentrated, but then we want to add in a platelet additive solution. So that's it. It's a number of reasons. One is there's less human product we're giving patients, so therefore we know it's safer. But also it means we then get that additional plasma, which we can use for other patients as well. So that's the study we're doing here. That's the first donor that's given it there. And that's a couple of the scientists who are actually based at Cambridge, having a look at the new platelets as well. This seems straightforward, but actually what's important to us is we get the component right. So has it got enough platelets in there? Is it going to meet the shelf life as well? Another bit of work we're doing is we need to make it simpler. Do you see what I mean? So we've got loads of blood groups, and actually we're trying to look to see if we can make our platelets and plasma universal. So rather than giving OPOS, A, neg and stuff like that, it's actually, can we strip that out and actually just say, if you want a platelet, you'll get a platelet. Can we make it a universal platelet? So this is another research trial we've got going on to see if we can make our platelets and plasma universal. Because what we've got is, in terms of hospital demand, we're really good with people wanting stock units sort of thing. But what hospitals really want is they want something that's completely universal for trauma or anything like that, or they want something that's really specific to their patient sort of thing. So they want to get to the extremes, which is what we're trying to do and deliver. Another component trial going on is actually funded by the military, and this is for dried plasma. So at the moment, you saw our plasma components, they've got 36-month shelf life, which is great, but if they're kept frozen at minus 40 degrees, when the army is out and about, they don't have a minus 40 freezer. Do you see what I mean? They could thaw something out, but there's more stuff they have to carry. There are commercial products available in dried plasma, but they're in glass vials. Glass vials are fragile, sort of thing. So actually what the ask was from the military was they want to have a dried plasma component that they can carry in their backpacks in whichever condition they're working with the view that actually if there's any military personnel that needs to get a plasma transfusion or a cryo transfusion, they can get it. There's no delay in that as well. Actually, that's another bit of work we're going on. This is going to go to clinical trials, and that's, again, the Cambridge team and Component Development Laboratory with our first unit of spray-dried plasma. The advantage for this is there's no glass in it. We've used our blood pack systems, and basically it's like a powdered plasma in there, and then all you do is you connect water for injection to it, you mix it up, and you can transfuse it straight away. We are expecting that once this gets trialled in the military, the civilian demand is going to have as well, because when people are in the operating theatres or dealing with emergencies, they don't want the delay of thawing that plasma. And actually, could this minimise that as well as a component as well? Debbie, do you want to come into this one?
No, that's So just to really just give you a little bit of information on plasma and where we've come since COVID hit us, really. So hopefully you'll probably have heard of both of these trials, the recovery and the remap cap trials. But when COVID hit, we were asked as an organisation, could we look at convalescent plasma and understand what the impact and effects is on patients in hospital, and whether it was going to make any difference at all to COVID patients and in their recovery. And although the outcome of those trials wasn't as successful as we wanted it to be, and it didn't necessarily have a massive impact on COVID patients, what it did was it kind of led to the re-looking at plasma for medicines as an organisation from that perspective. From the perspective of NHSB stopped making plasma for medicines back in 1990s, due to Creutzfeldt-Jakob disease, and obviously there was a concern and there was a risk of that being transmitted to patients who received a blood transfusion. But through all of this work, and you know what, I think one of the positives that COVID brought us was the re-looking at plasma and re-looking at UK plasma and what we could do there, and it's been really successful. So in February 2021, there was a lifting on the use of UK plasma to make immunoglobulin. I have to say that very slowly. So you know, that was a great thing for us, and the, you know, the information behind that is because that there've been very limited transmission through blood in that period of time since the ban had happened. So just to give you some kind of information around what happens in the plasma market at the moment. So that 7.4% is the annual global growth of the plasma for medicine market, and with the immunoglobulin as the main driver for that. 65% of plasma is currently collected globally from the US. So just to show that is the biggest area. And 0% of plasma is derived medicine manufactured from UK plasma since 1999. And that is something that we're looking to address now that that ban has been lifted. So immunoglobulin is a life-saving medicine, as you know, derived from blood plasma, and it's used to treat a large array of primary and secondary immunodeficiencies. Over 17,000 people in England are dependent on receiving this medicine, and many require it regularly every 6 to 12 weeks. And failure to maintain that supply of these patients can lead to chronic illnesses, significant reduction in quality of life or death. And it's probably has been a very under-talked medicine and life-saving treatment in this country. There's some lovely little pictures there of receiving their treatment. So some more key facts. As we mentioned, 17,000 patients in England. There's an annual expenditure of 400 million on that in England, because we buy it from abroad. An annual expenditure on albumin in England as well is 17.5 million, and 330,000 litres of volume of plasma is what we are targeting for NHSBT to collect annually. And our aim is to become self-sufficient for IG at 25%. So that really does kind of shift the global market that we're seeing. And that is phenomenal for the UK. I think that's a really fantastic achievement and ambition for NHSBT.
In terms of fractionation, so this is a bit different to blood component processing. So you get blood components, but this is a blood product. So this is under the medicines licensing. But how it starts is we get the plasma, it's frozen, but it goes through, rather than being more of a closed system process, it gets more into a manufacturing plant where you've got a plant and there's lots of stainless steel cylinders and piping and stuff like that goes through. And literally what happens is they receive it, they thaw the plasma at a controlled temperature, and then it generates cryo. The cryo not only has the coagulation factors, but has all of these proteins in there. And then it goes through the fractionation process, which is just saying protein purification. So it goes through chromatography columns, it goes through detergents, and basically, depending on the targets we want to do, we're going to split them out. At the moment, there's lots of stuff you can make from plasma as well. At the moment, the UK has got licenses to make IG and albumin as well, which is great because they're the two that are most in demand. When I started plasma fractionation processing all those years ago, you can actually make clotting factors as well for the haemophiliacs, but most of the medicine for haemophiliacs now is through recombinant protein production as opposed to human protein production as well. So once you've split it out, you then have to purify it and virally inactivate it as well. It goes through that process, and then it goes for a process of filling, and then it gets completely QC'd as a medicine batch product, and then it is released. So this is not something that we're going to start doing in-house. We would love to, but actually this is something that needs a big setup. And it's not actually NHSBT leading it. NHS England are leading this because they're the ones who buy the medicines for England and the UK. So what NHS England have done is they've commissioned NHSBT to collect the plasma. And one of the questions we're talking about is we can collect source plasma from our 3 sites, but about 98% of the plasma we collect at the moment is from recovered. So that is the plasma from that blood pack system that we've got on it. So we're redistributing it into the plasma for medicines process. So we collect it, and then NHS England have actually done a tender process, and they've awarded a contract to Octapharma, which is a large international pharmaceutical company. So once we get the units, once we've shipped them, it's all under NHS England's jurisdiction and contract. Octapharma gets it in Vienna. And actually, once they've done that, then they do some fractionations in spring, and then they send it over to Stockholm for filling they send it for purification and filling, and then it comes back to us in England. Now, we are expecting the first vial of IG to come back into England in January, which is great. And actually, that's a big thing, because then we're self-sufficient as a country. We're not relying on America to supply us those medicines as well. And like Debbie said, there's probably a lot more patients can have access to it as well. Apart from it being, with this process, we can strip out big profit gains as well, because it's going to be a lot cheaper to the NHS and a lot more available to the NHS as well. So that's something that's kicking off. I'm really excited actually. So hopefully in January we'll start seeing another first unit of IG coming in as well. And most of it is coming through our recycling of plasma units. So we kind of inside look of blood services, what the remit was, but actually what do we really need the inside of a blood unit, a blood process is actually you. You know, we need the donors to tell us what donor experience is. We need the researchers to tell us actually what's the best way we can do more for less. And actually we need people to share their experiences as well and research for actually enabling us to really do what's the right thing, because actually together we can actually really do what we want to do, and that's save and improve more patients' lives through blood. Thank you. We've got time for questions, haven't we, Anne? Sorry.
Absolutely. Yeah, absolutely.
Is there any questions in the room or online?
We have one online, but with Emmanuel, it's one from Netta O'Connell. I think she is a colleague at NHSBT, because I recognise the name. I'll read it out verbatim. Given that red cells are returned to platelet donors, how come whole blood donors aren't able to donate platelets in between whole blood donations? Are there any plans to change this restriction?
Oh, that's a good research title. If anyone wants to take it into the room.
Sounds quite.
Yeah. I mean, I think there's a couple of things. One is we probably do need the research, and how it tends to be is we want to get to a model where actually you're a donor. Do you see what I mean? So at the moment, people who are platelet donors are probably platelet donors because they are near a fixed site. Do you see what I mean? And actually, for someone who's not near a fixed site, it's really hard to travel to become a component donor. But most component donors tend to start off with as whole blood donors. I don't know. It tends to be, a lot of it tends to be what group they are. Do you see what I mean? Depending on what group they are, we want different things. For example, if you're an O-neg donor, we never want to lose you from donating blood. But if you're an A-neg donor, actually, we want you to donate platelet and plasma. Do you see what I mean? Especially for an AB neg, because AB neg is a universal group for the plasma components as only is for the blood. In terms of being interchangeable, I don't think we've done that, and really we would need some research for the donor safety.
I think it's part of the bigger piece of work that we're doing around the one donor journey. So actually there is a piece of work understanding how easy and how accessible we can make it, so that if you come in, we've got that new donor pathway, we understand what your blood group is, what from our side, what is the best product that you could give us from that blood group, but also for you, what is the best to do. And it could be that you donate platelets on one time you come in, the next time actually we need you as a whole blood donor, but all of that piece of work needs to be done, so we're not there yet.
It's for the safety of the donors.
Yeah.
Okay, great answer. Thank you.
Thanks, Guy. Excellent. Thanks. Really great presentation. Good to have an overview of what's happening and how things are changing. Couple of questions. One is about the post-donation testing. So it's good to see that some of the research we started a few years ago now coming to close to implementation. But my question is really about once you have the post-donation system in place, you get information about hemoglobin, but you can get much more information about the donors. And so is there any plan to think about how those information can be used, in relation to health of the donors? You don't only have, you have your hemoglobin level, but you can have all the food blood count. You actually you can plan to measure even more. Is there any plan to in that direction, looking really about the donor health side of?
Yeah, no, that's a really good question. And I have to say that as part of some of the discussions that I've had, we haven't got that far yet. We are in discussions around actually what are we going to be testing specifically. So for example, are we going to test more than the hemoglobin? Are we going to do the ferritin levels as well? But we haven't got to the point of actually could we get more data from those samples. But I think it's a really good question, and we should be looking at how we can join it all up and give more information.
Yeah, you're right. The other thing that comes ahead is when we start talking post-donation testing, they were like, actually, for some ethnicities, they've got lower hemoglobin anyway, right? So it's like, well, can't we just give them lower hemoglobin? It almost like if you've got too many problems to solve at one go, you get inertia sort of thing. You get pretty inertia. So like Debbie said, it's actually once we've done the trial for post-donation testing, then rolled that out, it's almost like you've got to, then do what's the next thing that's going to implement the research, keep the donors safe, and actually give us the best products as well. So there's going to be steps for it as well.
Thanks. I don't know if it's possible to put up the new improved post-donation slide up again. Mine, similar question. So it was just a couple of questions from in my case, from a donor perspective about that.
Is it this one or
the
actual process or session?
That one. Yeah. First one was, I think there are 3 questions, 3 points altogether. First one was, if something happens post donation testing, is there a risk that could sort of, in terms of the donor, that could sort of kind of put off the donor if they're told not to come back for 20 weeks or 26 weeks or possibly more, that having already donated, have you thought about what sort of consequences in terms of donor relations that might have?
Yeah, and again, it's a really good question, and that's part of doing the trial, so that we can really understand what happens at this point. No one should be tested with that post-donation test unless they've definitely got a really fantastic hemoglobin level. So certainly, what I would expect is those levels don't drop. So we take the sample and suddenly it's much lower than we thought. But there is, as part of all of this, we're looking at those discussions. What are we going to have? What are those discussions going to look like if it does do that? And we'll see that through the information that we'll be collecting. So the trial is going to be between 12 and 18 months long. So we'll have an awful lot of data to help us make sure.
Okay.
But we are thinking of all the potential risks and the communication to our donors as well.
Yes.
I mean, currently, we could have the same problem with the copper sulfate. You could pass that copper sulfate test
Yes.
and have a low hemoglobin afterwards. That, you know, we haven't seen much of that, but one of the reasons why we want to do the post-donation testing is the evidence suggests that it is a safer process for donors, so that they're not incorrectly having their blood taken from them, where potentially that could happen now. So we're trying to make it safer than our current process.
Just to comment on that, I mean, certainly where I go, which is the West End Centre, I mean, quite often, I think, related to age possibly, I find with the first pinprick test that quite often my blood is found to be sort of on the edge of, but then they do a second test in some sort of machine. So, I mean, is that not available?
Yes.
In other donor centres?
Yes, and that's available everywhere. We currently use a point of care device, which is a HemaQ machine, which is what you're talking about at the moment. It is available. Well, that is excellent. But what we're also looking at is, I suppose, is not necessarily gold plating it, but really making it the safest potential system that we have. And for a number of donors, even if we have post-donation testing, we'll still need to have that point of care device on session.
Yes.
Because the hemoglobin level might just be borderline and we want to check it at session, or there may be a new donor and that might be the only way to do that new donating. So we'll still have a secondary test on session to make sure that we can cover the bases. Okay.
And just very quickly, I mean, I noticed that in terms of the eligibility to return, that the time frame seems to be different from what it is at present, 12 and 16 weeks. Is that correct? And if it is, will that also affect blood supply issues?
Yeah, we're just looking through it at the moment. That the potential is we're making it pretty straightforward for this particular trial,
right?
Because we haven't done this before.
Yeah.
So we want to make sure that we're collecting that right information and understand the results. So we're doing it slightly different. The potential is in the future is that you could have completely different deferral rates depending on what your hemoglobin is. But again, this is the first step in us saying this is what we need to do at this stage of the trial.
Yes. And just the final point, I mean, if a shorter donation journey means, kind of less biscuits, I don't know.
Hopefully it will be more biscuit. It will be a much better donor experience, and definitely.
What we get as an organisation, right, myself and Debbie, we want to bring this change in like tomorrow sort of thing, but because we can't fail sort of thing. So a lot of our infrastructure is we have to have high resilience. So even projects like this and all our component projects is actually a lot of discussion about when. Because actually it takes out of the blood supply chain, could risk that. So a lot of it, I mean, if it's up to me and Debbie, we'd do something different every week, but it's about planning it and coordinating when we do it as well, which tends to sometimes take a little bit longer. Sorry.
Yeah, Yanning and then Neil. Yeah.
I was curious about your work on neurodiverse donors, right? So that's I think quite a recent thing. So I was curious about what if people who require easy read materials to understand, because I realized that for organ donation, you do have those available. You can say this is what it means to give an organ, why do you want to, but I haven't found any for blood donation. So I wondered, is that something that you're thinking about implementing? Because that could be, I guess, making it more accessible to people who have learning difficulties or whatever it is.
Yeah, again, I mean, the neurodiverse piece of work that we're doing at the moment from a donor perspective is completely new, but we have been looking at that journey from an organisational perspective for a long time, and it's quite complex because easy read materials for someone who's dyslexic is potentially potentially different to someone who has ADHD, or there are different.
Oh, sorry, I mean the one where they have like images alongside the words and then a shorter kind of sentence style and those kinds of things. Where specifically for people who have learning difficulties, not ADHD or neurodiversity.
Okay, so not neurodiversity, more learning difficulty.
Yeah.
So again, we have been looking at that. We have had some significant input from both MenCap and other organisations to help us to support learning difficulties and learning disabilities through the session. So it's just making it it's making it specific and right for the individual, because each individual challenge will be different. So rather than having something generic, it's trying to make it specific for those donors coming through.
I see. And I just have a final thing, which is I'm curious about the safety of donation for people who, let's say, are wheelchair users, and let's say they're able to transfer themselves to the donation chair, right? But then, for example, for applied muscle tension, where usually you kind of apply the tension to your lower body, right? If people can't do that, there might be an increased risk of like VRs and things like that, right? So is there any kind of work on what other methods you can use to engage in those kinds of, like I said, the people who cannot use that part of their body, let's say?
Yeah, no, that isn't my area of expertise. This is where I would normally call on someone like Ella, who's our chief nurse for blood supply. So I need to take that question away to her and get that fed back to you.
Right. I know, Neil, you have to leave around.
I-
So if you take,
Cancelled that.
oh, no, that's good.
I can grab it on as long as you like.
We have 2 questions online, so I'll take Neil next, and we have 2 questions online.
Okay, great. So firstly, your plastic bags. Can't you get a medical use exemption for that chemical?
So
You should be.
it's a really good point. So at the moment, it's on its medical use exemption. That sunset period's going to set sort of thing. So as that I think that lysate material that's in part PVC is only in medical devices at the moment because it's got the exemption for the sunset period. That exemption is finishing, I think, around 2027, but it's for Europe. But the other thing is, well, so we could, technically, we don't need to follow that because we're outside of Europe. However, most of the blood bank manufacturers want to cater one product.
Yeah.
See what I mean?
Yes.
So actually, it's not an option. To see what I mean? If some of the suppliers are changing, we need to pivot to it. The challenge we've got is, at the moment, the plasticizers we've got at the moment are quite consistent across all blood pack manufacturers. With this change, they're using different ones. Okay, now that's really challenging for us because we've got to validate our components for all of those ones as well. But that's something, it's a big headache.
And I think last time I sat in one of these was the guy from Denmark explaining.
Christian,
Who explained how he'd been doing genetic screening and to see whether the donors could pass anything on.
Yep.
And he'd been writing to people saying, we don't want your blood anymore. Do you do that genetic
So
screening?
we do. So there's a couple of things. So I didn't cover it here, but we do genomics testing. So at the moment, we are testing all sickle cell and thalassemia patients in England. So, all hospitals can send us a sample and we're doing a genotype for those patients so we can better match them. What we then want to do is we want to start genotyping donors or the red cell antigens I'm talking here in terms of, I'll come back to the safety one, so we can have better match for them as well. So at the moment we're doing patients, we're having discussions, like one of the machines we brought in, I never saw, is we're bringing discussions at what point do we pivot from serology testing, which is just seeing what's on the red cell, to doing the genotype testing as well. So we've got that capability. We're not doing it as standard yet, but also the work the BTRU's done on STRIDE donors, you guys have got a lot of genotype information there as well, is how can we use that? So we're having discussions literally now on how can we bring that data in so we can better match it. In terms of the blood safety, transfusion transmission and stuff like that, there's another BTRU for genomics and microbiology screening going on, and they're working on exactly that as well. And we're working with the people within NHSBT on the serology side to see what can we do, because the similar technology is being used for different reasons, so we're bringing them together.
Great. So that's lovely. 2 questions online. My battery's running low. Better hurry up here. The first one is from Nicole Stout, who's our program manager for the BTRU, and she's asking, does Optopharma do any testing on UK plasma?
So they test on a finished product. So we screen the plasma, and we've actually introduced a new screening test because it's applicable for plasma for medicines, which is HIV, so hepatitis A and papillomavirus B19 as well. So we brought that testing in-house because we want to make sure they screen negative when we give it to the fractionator, because we don't want that to be the reason we lose a pool. So what they do in fractionation, they pool all the donations together before they make it. The fractionators, they will be testing final pool products or the things that they've got to test for, as opposed to individual donors, which we do. So they'll be testing for the product as a medicine at the end, as opposed to screening.
Yeah. Okay. that definitely makes sense. So 2 questions from online listener Rachel Ossie. One is, she says, thanks for the presentation. What strategies are in place to prevent shortages during times of increased demand emergencies? That's a very broad question. And the second one is about a friend of hers who has donated blood, discovered in the process she had malaria antibodies, which means this person can no longer donate. For individuals like that who are enthusiastic about contributing but are permanently deferred, are there any current strategies or innovations to allow them contribute, such as donating for research or specialised uses? Okay, yeah. Battery's gone, that's the end of that. So yeah, the first one is very broad, which I'm sure you're asked a lot about shortages.
So at the moment, we're in Amber Alert, which means, we're close to being shortage for certain blood groups as well. There's a lot going on. So first of all is we have a stock that allows us to respond to things as well. And then actually what we do is we work closely with the hospitals. Mm-hmm. So actually the hospitals can reduce their stock holding or limit their use, actually, so we can serve things over away. And actually it's about the donor engagement. It's always having that new supply of new donors to replace those that can no longer donate. And it's the challenge that we have most discussion about. Debbie said we're opening more donor centres, and that's a response to resilience. We want to be more resilient. Is there anything else on that?
Yeah, so I mean, if I could share with you all the strategies in place, it's just massive. Yeah. So for every possible scenario, I think there's some very generic responses that we do, which the hospitals will be aware of, and then there's all the activity behind it. It's massive, but we also look by components. So I'm leading on a piece of work for the platelet stock resilience, and it's all the activities that we can do to support that. Mm-hmm. And actually, what is the problem that we're trying to overcome, and really understanding the problem. So there's lots of different activities to get us to a point of being as resilient as we possibly can, and some of them are focused on our donors. You know, how do we encourage our donors to come in when we need them at the right time? How do we ensure that we have the capacity that Aman said, so more donor centres, whatever that is, so we have places. And then there's a big resilience piece around our operating staff as well, and making sure that we have the right people to be able to take the collections as well. So lots of different,
very complex,
very complex,
things, planning. And then for people who are not allowed to donate for various reasons like malaria, but others, any options for them?
So at the moment we asked to be an advocate, as in get other people to donate as well. But actually, I can't remember who I had a conversation with, but it wasn't too far off the lines. Actually, can we just get people who blood will not be screened for patient use? Can they give it for research use? Which will probably be possible. We don't do it at the moment because we've got consent for the whole processes as well. But the other thing to note is we're becoming a warmer country. Do you see what I mean? Which is really good, I guess. Sometimes, we can more nice weather, but actually what that means is we might see disease prevalence come in that we're not used to. Do you see what I mean? Yeah. Such as malaria and other things as well. And actually that's something that's part we're doing at the other BTRU is actually monitoring what more do we need to screen, which is good for the safety, but it actually means we're then taking more blood out of the system as well. So there's probably things we've got to do there. But at the moment, unless Debbie, I'm wrong, we can't accept a donation that we can't give as a final.
No, it depends on the reason for deferral as well. So whether it's something like malaria or actually it's a wider health implication. But I know that the donor experience team are looking at widening what can we do, to get the advocates from a donor perspective, because it tends to be donors who bring other donors as well. But what else they can do on a bigger piece, and then also in the same way that we reward donors for coming in, for giving donations, can we reward people who support the service as well? Because that's a really good motivator.
So it's a bit of, if you have a health implication, another blow if you can't. One of the things you like doing is donating blood. I guess just answering from a university point of view, there's lots of stuff going on in universities. Maybe there is a chance to help a local university or some kind of research in a hospital with your blood, if it's not, assuming all blood has to be considered infectious, doesn't it, until it's tested. So that's great. I mean, that was a terrific discussion. I think we probably could spend longer. I want to thank our 2 guest speakers, and we have the room till 3. So in discussion with everybody's welcome to stay for a discussion, assuming that you still have time to stay with us till then. And thank you very much for traveling the distances to be with us today. And thanks to everybody online too.